Today we shall talk about mitochondria. So far, we have talked about organismal aging, only to start digging deeper and deeper all the way down to the cellular level in order to explain some of these organismal aging phenotypes on that cellular level. But let's go even deeper to the subcellular level and investigate subcellular compartments, the organelles, and how they may influence aging. The most important organelle here is the mitochondrion, which we already briefly talked about in Module 4 when we discussed the role of stress in aging. Mitochondria are often called the power plants of the cell, and while it may sound a bit cliché, there's really no better way of describing them. Mitochondria perform the majority of ATP-generating reactions, including the Krebs cycle within their matrix and oxidative phosphorylation at their inner membrane. Thus, the catabolism or breakdown of biomolecules from amino acids to sugars and fats takes place within the mitochondria. As we have seen before, mitochondrial metabolism leads to the generation of reactive oxygen species. All this makes mitochondria very interesting to the field of aging research. Mitochondria used to be free-living prokaryotes that were eventually engulfed by other prokaryotes. The two cells then entered into a symbiotic relationship. This is called the endosymbiont theory and describes the origin of eukaryotic cells. For the mitochondrium, it means that it has two membranes, an outer, relatively permeable one, and an inner, impermeable membrane. This inner membrane is the site of the electron transport chain and oxidative phosphorylation where ATP is generated. The inner membrane encloses the mitochondrial matrix, where other metabolic reactions are executed, including the Krebs cycle and fatty acid oxidation. The inner membrane is highly invaginated. It forms folds, or cristae, to increase its surface area and to therefore increase the efficiency of ATP generation. During aging, invagination becomes less and less pronounced, and the shape of the inner membrane changes from cristae to tubes and finally to rounded vesicles. Interestingly, and a remnant of their bacterial origin, mitochondria contain their own DNA, which exists as a single circular chromosome within the matrix, similar to the nucleoid in bacteria. Mitochondria also have their own ribosomes, which resemble bacterial ribosomes more than they resemble eukaryotic ribosomes. Moreover, mitochondria reproduce according to their own reproductive schedule, independent of that of the host cell. Mitochondrial reproduction occurs by fission, the way bacteria reproduce, and not the way eukaryotic cells reproduce, which is mitosis. Finally, contrary to what most textbooks show, mitochondria exist within the cell not as perfectly ellipsoid small organelles, but rather as elongated, highly interconnected structures that may even form a filamentous network. This is the direct consequence of their own reproductive activity, which means new mitochondria are continuously generated through fission from existing mitochondria or are fusing back together to form a larger structure. This network is highly dynamic and responds to the metabolic needs of the host cell. Even though mitochondria contain their own DNA and reproduce independently of the host cell, they are highly dependent on and in constant communication with the cell's nucleus. Mitochondria generally only contain the genes for their own translation system, including the tRNAs and rRNAs, as well as the genes for the enzymes of the respiratory chain. All other genes have been exported to the nucleus and need to be imported back by specialized transport proteins and chaperones. These are proteins that aid in the correct folding of proteins. These nuclear encoded genes are transported through a set of transmembrane transporters called TOM and TIM in an unfolded state. Only after translocation through this membrane pore will chaperones then aid in the correct folding of the transported proteins. Mitochondria also rely heavily on metal ions for the redox reactions carried out during metabolic processes, including copper and iron. A separate set of chaperones is responsible for this iron transport and delivery to the mitochondria. They furthermore keep these reactive molecules inactive until they reach their final destination. Recently, it has been shown that the components of these transport mechanisms may influence longevity, which highlights the role of mitochondria in longevity, apart from the ROS generation. The exact mechanisms of that, however, remain unclear. As we mentioned earlier, mitochondria continuously undergo fission and fusion events. During aging, the homeostasis of these events gets disturbed, which leads to more and more fragmented mitochondria, as well as less tubular and more nuclear-oriented mitochondrial networks. In addition, morphological changes of the inner membrane lead to an increased loss of cristae and a more tubular or vesicular inner mitochondrial membrane. This is thought to decrease mitochondrial function and efficiency. It has been shown that deletion of the genes responsible for these fusion events, called DMN1 in yeast or DRP1 in mammals, can drastically alter longevity. However, it is also required for proper function of mitochondria-initiated programmed cell death or apoptosis, which plays an important role in tissue development and remodeling in multicellular organisms. Thus, DRP1 may be another example of an antagonistic pleiotropic gene. Furthermore, mitochondria undergo a process called mitophagy, or mitochondrial self-digestion. This is similar to the process of autophagy, a mechanism by which damaged cells self-destruct by self-digestion. Mitophagy is therefore important for maintaining a healthy and functional population of mitochondria, especially by removing mitochondria that are unable to fuse back onto the larger network. Dysregulation of mitophagy, or loss of its efficacy, may thus further contribute to aging phenotypes. The main function of mitochondria is to produce ATP via the citric acid cycle in the matrix and oxidative phosphorylation via the enzymes of the electron transport chain within the inner mitochondrial membrane. The electron transport chain consists of 4 large multi-protein complexes that take electrons from the reduced metabolic intermediates NADH and FADH2 and transports them to the final electron acceptor, which is oxygen. The energy contained in these electrons is used to pump protons across the inner mitochondrial membrane to create an electrochemical gradient. In turn, this electrochemical gradient can be used to drive a molecular motor called the ATP synthase, or complex 5, to drive creation of ATP molecules. Along this way, electrons combine with molecular oxygen under very controlled reaction conditions at complex 4 to form water. Unfortunately, electrons and oxygen are highly reactive species, which means that the electron transport chain may produce inefficient side reactions, during which electrons may react with oxygen at other places along the ETC, notably complexes 1 and complex 3, but not form water, but rather highly reactive superoxide anions. As discussed earlier, superoxide can be converted to hydrogen peroxide or even hydroxyl radicals, all which constitute reactive oxygen species. These reactions form the basis for the free radical theory of aging. Since reactive oxygen species are highly reactive molecules that can easily react with and therefore damage biological macromolecules, detoxification of these molecules into harmless water is critically important for a cell. As discussed earlier, these detoxification mechanisms include SODs, catalases, and peroxidases. Conditions that slow the flow of electrons throughout the electron transport chain lead to an increased lingering, if you will, of the molecules that carry the electrons throughout the individual electron transport chain complexes. This, in turn, increases the likelihood of those electrons to react with oxygen to form superoxide. Thus, damaged mitochondria with inefficient electron transport create more reactive oxygen species. Conversely, conditions that speed up electron transfer reduce the probability of ROS creation. For example, by inserting a wide channel into the inner mitochondrial membrane in the form of a protein called uncoupling protein, or UCP, electron flow is uncoupled from powering the molecular motor of the ATP synthase. This means protons can simply flow back into the matrix via the UCP channel, which is easier than flowing through the ATP synthase. This speeds up the electron transport chain, reduces ROS generation, and can extend longevity. Interestingly, mitochondrial uncoupling is the mechanism by which brown adipose tissue creates heat and regulates body temperature. It is no coincidence, therefore, that researchers have found that brown adipose tissue is the healthy type of adipose tissue, as opposed to white adipose tissue, which is the main fat storage organ, an excess of which leads to the detrimental health effects associated with obesity. Thus, uncoupled mitochondrial membranes, or leaky mitochondrial membranes, reduce mitochondrial membrane potential and reduce ROS generation. The downside of this is that mitochondrial ATP generation is directly correlated to its membrane potential. The higher the potential, the more ATP can be produced by ATP synthase. Once again, highlighting the trade-offs that are built into the system and that are often associated with longevity. While the most prominent, the electron transport chain is not the only source of ROS generation within the mitochondria. Many metabolic enzymes contain metal ions that are particularly susceptible to reactive oxygen species. In the example shown here, the enzyme isonatase contains what is called an iron-sulfur cluster. As the name implies, the enzyme contains a cluster of iron and sulfur atoms that play an active role in catalysis of this enzyme. Isonatase is the second enzyme in the Krebs cycle. When ROS react with the iron-sulfur cluster at the center of this enzyme, they change its reactivity and in turn produce even more ROS, further exacerbating the damage. As the active site of isonatase thus gets damaged by ROS, its catalytic activity drops, leading to a decrease in the metabolic flow through the Krebs cycle, which in turn leads to reduced ATP production, which in turn could result in cell death. With all this damage emerging from mitochondria and in turn acting directly on mitochondrial macromolecules, it is imperative that mitochondrial damage is contained and repaired, or if that is not possible, to remove damaged mitochondria altogether. This mitochondrial quality control system acts on several levels, the first of which is to scavenge any ROS that may develop. Any damage that still occurs may then get repaired, but if that is not possible, mitochondria may simply fragment away from the network and undergo mitophagy. If the damage to mitochondria becomes too extensive and no intact mitochondria remain, mitochondria then may trigger cellular suicide. We already talked about the ROS scavenging systems in the context of cellular stress. Superoxide dismutase, catalase, peroxyreductase, all these enzymes work in concert to convert ROS into harmless water. While it is an attractive idea, as we have already discussed earlier, simply increasing the amount or the activity of these enzymes does not lead to beneficial effects on longevity. And similarly, removing these enzymes altogether often does not even shorten lifespan, although it has been shown that this contributes to various disease pathologies. Even the best scavenging system simply can't catch all these damaging molecules, leading to the accumulation of mitochondrial damage. When mitochondrial proteins are damaged, a likely consequence is a decrease in ATP production, which we have discussed earlier. Since proteins usually can't be repaired, mitochondrial protein quality control consists of the removal and replacement of damaged molecules. Damaged molecules in the outer membrane will be removed by the cytosolic repair and removal system, called the ubiquitin proteasome system. We will talk more about this system when we discuss protein homeostasis in a later module. Proteins of the mitochondrial matrix are degraded by the protease LON, while proteins of the inner membrane are targeted by a membrane-bound protease called MAAA. The breakdown products are transported out of the mitochondria into the cytosol, where they are further metabolized and recycled to be made available for protein synthesis. LON and MAAA belong to the same class of AAA proteases. Removing LON leads to cells with abundant mitochondrial damage and extensive mitochondrial dysfunction. LON overexpression in turn extends longevity in yeast. In fact, aging is often associated with decreased LON levels. When LON is overexpressed, not only is longevity increased, but also mitochondrial function, including reduced ROS generation, increased ETC efficiency, and decreased levels of damaged proteins, as measured by the amounts of protein carbonyls, a form of protein damage commonly associated with aging. These data clearly demonstrate that activation of mitochondrial protein quality control systems counteracts mitochondrial damage that is accrued over time. Interestingly, it is an intervention on the organelle level that has wide-reaching effects on the whole organism. Another class of proteases with a clear connection to aging and lifespan are the small heat shock proteins, especially the mitochondrial small heat shock protein 22. Overexpression of this protein has been shown to increase longevity. However, while this protein may function as a chaperone, the mechanism of its lifespan extending properties still is largely unclear. If mitochondrial damage becomes too severe, damaged mitochondria may be removed by mitophagy. This process may involve the interplay of two proteins called pink and parkin, where pink is able to recognize damaged mitochondrial membranes, which in turn then recruits parkin. Parkin then is responsible for sending the damaged mitochondrium to the lysosome, where it is digested. Parkin in turn then is responsible for sending the damaged mitochondrium to the lysosome, where it becomes degraded. Interestingly, parkin is thought to be directly involved in the development of Parkinson's disease, a neurodegenerative disease that is closely associated with aging. Parkinson's disease, thus, is a disease of mitochondrial dysfunction. If ROS scavenging, damage repair, or even mitophagy can't remove or inhibit the damage, the only option that is left is to remove the cell altogether that has the damaged mitochondria. This process is called programmed cell death or apoptosis. Apoptosis is also an important developmental process that plays a role in tissue remodeling. In mature organisms, as discussed earlier, apoptosis is necessary as a protective mechanism to remove damaged and potentially dangerous cells from the organism. As such, it is crucial for the control of hyperproliferative cells, such as cancer cells. The flip side is that too much apoptosis may contribute to the aging process by removing too many cells. Apoptosis can be induced by an extrinsic and an intrinsic pathway. The extrinsic pathway is induced by external signals that directly leads to the activation of a class of enzymes called caspases. These caspases digest cellular proteins, which in turn kills the cell. The intrinsic pathway gets activated when mitochondria rupture and release their context into the cytosol. This rupture can be triggered by a depolarization of the inner mitochondrial membrane and/or the opening of a pore within the inner membrane, often caused by cyclophilin D. This leads to osmotic lysis of the mitochondria and the release of proteins into the cytosol that usually are not found there. For example, cytochrome C. Another molecule is called AIF, or apoptosis-inducing factor. These molecules, in turn, once again, are capable of activating the caspases, which then, of course, leads to the destruction of the cell. Thus, mitochondrial function not only affects energy metabolism of an organism, but it's also directly linked to tissue homeostasis and health. Check your understanding. Describe mitochondrial biogenesis and homeostasis. Explain mechanisms of mitochondrial ROS and RNS generation. Discuss mitochondrial quality control and the consequences of its failure.